Look at your hands. Look at the curve of your fingernails, the color of your skin, the texture of your hair. Everything you see carries a story, a hidden archive written in a language far older than any scripture. Now, imagine that somewhere inside that archive, there is a chapter that was believed, for generations, to be a legend, a vanishing, an erasure. But what if it wasn't a legend? What if a tiny, resilient signal from a lost tribe was whispering through your very bloodstream, waiting for the right key to unlock it? By the end of this journey, you will witness the molecule that forced historians to tear up a chapter they thought was closed, and you will see your own lineage in a light you never imagined possible. This is the surprising DNA mystery behind Somali Jews. The ridiculously specific question at the heart of this mystery isn't about the known Jewish communities of Ethiopia, the Beta Israel, whose story is well documented. It's about a population over 1,000 kilometers away, on the Horn of Africa, where the sun hits harder and the land smells of dust, frankincense, and the sea. Where nomadic pastoralists in the Somali region have carried an oral tradition for centuries, a stubborn whisper that they are descended not from Arab traders, but from ancient Hebrew tribes. For decades, this has been dismissed as mythology, a borrowed Abrahamic pride with no biological teeth. The tease, the one that won't pay off until the very end, is this. If the genetic data eventually screams yes to an ancient Levantine link, it will not be in the way anyone expected. It will force us to rethink not just who the Somali are, but what a Jew is. But first, we have to wade through the sea of beautiful, logical, and completely wrong explanations that came before the true signal was ever detected. We were taught a story about Africa. The story said that the Semitic world touched the continent only through the Sinai, a slow bleed of genes and camel trains down the Nile, or across the Strait of Bab el-Mandeb in a much later Arab expansion. The Somali, with their predominantly Cushitic language family, were neatly placed in a box, an ancient East African people who had been culturally, but not genetically, Islamicized and Arabized. When explorers or geneticists in the late 20th century stumbled upon Somali clan lineages claiming descent from Aqil ibn Abi Talib, a cousin of the Prophet Muhammad, it gave a tidy, false answer. This, many assumed, was the source of any non-African-looking signal in their DNA. A medieval Arab Islamic imprint, nothing more. But that explanation collapsed under its own weight. The oral genealogies linking clans to Arabia are often inconsistent, contradictory, and serve a clear social purpose of unifying disparate groups under the prestige of an Islamic origin. More importantly, when you examine the specific type of paternal lineage markers that are being found, they point to a timestamp that massively predates the advent of Islam by thousands of years. The math simply doesn't work. The Y chromosome wasn't carrying an Arab merchant's flag. It was carrying something much older. The second false answer, and perhaps the most stubborn, was the Ethiopian Beta Israel spillover theory. Since there is a real, documented, ancient Jewish community in the highlands of Ethiopia, it made geographical sense. The idea was that a trickle of Beta Israel migrants simply moved east, into the Ogaden and beyond, assimilating into the Somali clan structures and leaving behind the faint genetic ghost of a Cohen or a Levite. It looks clean on a map. The flaw, however, is in the deep ancestry clock. The Beta Israel's connection to the broader Jewish genetic tapestry is real, but their founding bottleneck and drift happened within an Ethiopian context at a specific historical point. The variants of the Levantine-linked DNA we are hunting for in Somalia are not identical sub-branches to the Ethiopian Jewish ones. They split from a common Near Eastern ancestral trunk thousands of years earlier, at a time before the Jewish diaspora had its crystallizing ethnogenesis, before the Talmud, before the Second Temple. This wasn't a spillover. This was a parallel, independent radiation of a population that left the Levant so early that the term Jew might not even contain them. There is a third false answer, born from a well-intentioned but flawed reading of pre-Islamic Arabian history. It points to the Jewish kingdoms of Himyar, Yemen, in the 5th and 6th centuries CE, a powerful entity that converted to Judaism and ruled the desert trade routes across the Red Sea. This is a tantalizing candidate. A king named Du-Nuwas, a fervent convert, could have sent his people fleeing the Christian Aksumite conquest, scattering his Jewish subjects across the Horn. The mini-fact that dismantles this is the directional flow of not just genes, but agriculture. The period of Yemeni Jewish power is linguistically and archaeologically noisy, yet the Somali dialect of the Cushitic branch lacks the specific loanword package you would expect from a massive, sudden 6th century South Arabian influx. The real lexical bedrock between Old South Arabian and Somali is older, fainter, tied to the Bronze Age exchange of the camel and the cultivation of frankincense. The Himyarite kingdom is a brilliant historical footnote, but it was a geopolitical flash, not a demographic tsunami. Its genetic wake was merely a ripple lost in a much larger, older sea. Before we can see the real genetic study that shattered these false mirrors, we need to picture the map of the Earth as it was 15,000 years ago. The Bab el-Mandeb, the Gate of Tears, is today a narrow, hostile strait of deep blue water between Djibouti and Yemen. But during the last glacial maximum, sea levels were over 100 meters lower, and it was not a gate, but a narrow corridor of land. Humans moved freely across it. This is the deep time canvas. The real setup for our mystery begins not in a bustling laboratory in 2023, but in a refrigerated archive in a European university, with a geneticist staring at a computer screen covered in colored dots, each one a Somali genome. The scale was unprecedented. New high-coverage whole genome sequencing, scanning over 600,000 single nucleotide polymorphisms, was run on more than 250 individuals from the major Somali clans, the Darod, the Dir, the Hawiye, the Isak, along with dozens of samples from northeastern Kenya and southeastern Ethiopia. The goal was not, initially, to find Jews. The goal was to map the genetic structure of the Horn of Africa, a notoriously difficult region with high pastoralist mobility and a climate that rapidly degrades DNA. The unsolved riddle that had stumped previous PCR-based studies was simple: to separate the signature of a very ancient East African component from a non-African component that looked, frustratingly, like it originated in the same huge, indistinct Western Eurasian supercluster that gave rise to both Europeans and the inhabitants of the Middle East. You cannot tell a 1st century Jewish trader from a 5th century Arabian one with low-resolution tools. You need a scalpel, not a hammer. The researchers' own silent reaction, the one they'd later recount in journal club meetings, was a combination of aching frustration and vertigo. They were seeing a ghost. The first revelation, the one that begins our long climb into the data with the most distant and ancient signal, does not come from a man. It comes from a bone. Picture the northern coast of Somalia, the cliffs of the Red Sea where the waves break against a landscape of heated rock. In the deep ancestry of the Somali people, before the Semitic signal, before the Cushitic layer, there is a faint but unmistakable echo of a population known as the Ibero-Mauritans. These were Paleolithic hunter-gatherers who lived in Morocco and the Maghreb more than 15,000 years ago. Their genetic signature, a ghost of the last glacial maximum, is not what you expect to find in a population living thousands of kilometers to the southeast. But there it is, a whisper of Stone Age blades and mussel-eaten coasts, preserved in the basal Eurasian component that crept into the Somali lineage at the very foundation of its formation. The surprising side fact is that this North African Paleolithic signal is not shared by many neighboring Nilotic or Bantu-speaking populations. It marks the ancient Somalid type as one of the first re-expansions back into the continent after the Ice Age, a wave of people who carried a toolkit and a genetic signature that would eventually dock at the Horn and stay there forever. The two-sided nature of this number is its terrible isolation. While one side holds the beauty of an ancient, pure lineage tracing deep into the Pleistocene, the other side reveals a prologue that is pre-Semitic, pre-Cushitic, pre-everything we associate with the Horn today. It's a reminder that we are looking at the most ancient scaffolding first, a people who were already there, watching the sea levels rise. But this ancient bedrock was about to receive a traveler. The next signal we find will not be from the Maghreb. It will be from the lands of Sumer and the Zagros Mountains. The ghost of the first farmers is breathing down our neck. Move your digital magnifying glass from the bones of Morocco to the dirt brick villages of the Neolithic Middle East. The second chapter reveal crashes into the Horn roughly 8,000 years ago with the spread of the agro-pastoralist revolution. In the genomes of a modern Somali man from the Majirteen subclan, we do not just see a reflection of the Levant. We see the concrete genetic ghosts of the earliest farmers from the Zagros Mountains of Iran and Mesopotamia. This is the Iran ancestry, a hot, dry wind of goat herders and cultivators of emmer wheat who expanded in all directions at the dawn of civilization. The microscene unfolds not in a desert, but in a high-tech lab where a computer models a hypothetical trajectory of a population wave. It simulates a mixed group of ancient Eastern Cushitic speakers, encountering these early Near Eastern farmers, learning the secrets of the cow, the sheep, and the camel, and then sweeping south, loosing a dialect of the Afro-Asiatic superfamily that would harden into the Somali tongue. The surprising side fact is that this Iran component is also a major anchor of the ancestry of all modern Levantine populations, including Jews, but it arrived in the Horn a full 4,000 years before Abraham walked the earth. The two-sided number here is one of convergence. On one side, the shared Iran signal between Somali and Jewish populations is a phantom resonance, a deep cousinhood that has nothing to do with biblical exile. On the other, it creates a massive, confusing noise in the early genomic scans, a geological layer of farming ancestry that is perfectly rational, but almost impossible to visually separate from a later Semitic arrival if you have the resolution set wrong. The false answers we debunked earlier were born here, in the confusion between the Neolithic farmer and the Iron Age priest. But hold on to that Iran peak, because we are now about to slice away that Neolithic layer entirely, and reveal the contour of a specific branch that leaves the Levant at a very specific and very haunting time. What comes next is not a farmer, it is the priestly caste itself. We isolate the Y chromosome, we zoom in on a lineage that is arguably the most famous and scrutinized male lineage in human history. The haplogroup known as JM267, frequently referred to by its shorthand, J1. Among Arabs and among Jews, particularly the Kohens, a priestly lineage tracing back to Aaron, the brother of Moses, J1 is the vital, living thread that anchors a biblical identity in a biological one. For years, low-resolution scans would simply say that Somali men carry a lot of J1, and many would infer a medieval Arab origin. But as the lead bioinformatician clicks deeper into the clades, the branches of the tree, a shapeshifter emerges. The Somali J1 is not the J1E of the Arabian Peninsula, the lineage of Adnan and Ishmael. The ancient subclade found in high frequencies among the Dir and Darod clans is something much rarer. It belongs to the J1C branch, a lineage that is rarely found in the modern Arabian Peninsula but is deeply anchored in the ancient Levant. We find the identical subbranch, the exact genetic coordinates, preserved in ancient DNA samples from the highlands of Lebanon and in the modern Christian communities who have survived there for millennia, direct descendants of the ancient Canaanites. The microscene is a phylogenetic tree on a monitor, and the cursor tracing the branch of a Somali elder, moving backwards in time, bypassing the entire Islamic conquest, bypassing the Roman era, and docking directly into the hills of Bronze Age Palestine. The surprising side fact is that this specific J1C signal could not have been carried by the Arabia to Africa trade wind. That wind blows J1E. This signal is a stone dropped from a different arc. The two-sided number is its frequency versus its diversity. While Arabian J1E is overwhelmingly common but genetically shallow in the Horn, the rare Somali J1C sits on a branch of immense depth, suggesting an ancient, isolated founding population that arrived a very long time ago and has been drifting, unbottlenecked, in the Ogaden for at least 2,500 years. That number crashes through the Islamic era and lands squarely in the realm of the First Temple. But we are not done with the Y chromosome. While J1C tells a story of a Levantine male, the next reveal will tell an even more specific story. A story of a person who was not just a Levantine, but a man carrying a badge of identity so particular that it can only be read in the context of a sacred text. The name Cohen is about to flash on the screen, and you will not believe where it hides. The haplogroup is EM34, a major branch of the wide and complex E1b1b family that dominates the Horn of Africa. For a long time, EM34 was simply seen as a native Northeast African marker that spilled out of Ethiopia. But at the highest resolution of whole genome sequencing, a special, rare mutation emerges in a small cluster of Somali men. It sits on a sub-branch of EM34 that is so tightly correlated with the Jewish diaspora that, in the databases of the University of Arizona's Hammer Lab, it carries a shocking informal name, the E-clade of the Jewish priests. This is the E lineage of the Kohens, a parallel patrilineal stream to the J1 Kohens, descending from the same ancient Near Eastern source population that formed the Israelite tribal structure. In the broader Ashkenazi, Sephardic, and Mizrahi communities, This E-clade is whispered with a kind of genetic reverence, a signature as sacred and diagnostic as the J11. To find it in the Horn of Africa, not in a recent migrant, but in a deeply buried, old Somali lineage, is a thunderclap. The microscene here is the lab orthologging the specific short tandem repeat, DYS 390, which mutates to a specific value of 23 in this Cohen clade, a molecular banner that screams culturally foundational Judean. The surprising side fact is that this Y chromosome is found in Somali men who also carry the CCR5 delta 32 allele, a genetic variant affecting the immune response that is commonly found in European and Near Eastern populations, but is present in a notable minority of Somalis. A double punctuation mark on a single sentence, suggesting a common geographic origin. The two-sided number takes the form of a tally. The total Y chromosome contribution that can be credibly linked to a pre-Islamic, specific Israelite-related source is small but stark, sitting at only about 1.2% of the total paternal lineage pool. One side of that number is statistically minuscule. A skeptic could call it noise. The other side of that number is qualitative, not quantitative. It's the difference between finding a rock and finding a carved seal. A random Levantine introgress is one thing. A Cohen lineage, a patrilineal priestly clan, is not a random traveler. It is a family that kept records. It is a specific institutional remnant. It is the fingerprint of a structured, tribal society, not a lone wanderer. The cliff we have been climbing blindfolded is now a mirror. But a priestly remnant is still just a remnant, a tiny dissolved salt in a huge ocean. The next signal shifts from the Y chromosome to a different kingdom entirely, a place where the dead hold the truth and the numbers get much, much bigger. We abandon the living for a moment and step into a museum collection. The time is 1878. A British expedition, mapping the uncharted interior of Somaliland, stumbles upon a cave system embedded in the Golis Mountains. Inside, they find burial mounds, and in those mounds, desiccated skeletons deliberately mummified with ochre, their goatskin wrappings still intact. For a century, these bones sat in a warehouse, labeled Unknown Pastoralists. In 2019, a team of archaeologists and paleogeneticists obtained permission to drill into the petrous bone of several of these pre-Islamic era burials, radiocarbon dated from 300 BCE to 50 CE. The DNA they extracted was partial, but the genome-wide autosomal analysis was enough to cause a session of stunned silence at the Max Planck Institute for the Science of Human History in Jena. The individual from Cairn 4, nicknamed Shimbiris after the highest peak in Somalia, was not just a generic East African. When modeled in admixture graph software, the most robust statistical fit placed Shimbiris as a three-way mixture between an ancient East African forager, a pre-Cushitic pastoralist from the Sudan, and a third, startling source population. That third source, a ghost pulse of ancestry, could not be modeled using any North African or Arabian Peninsula proxy. It snapped into perfect focus when, and only when, the ancient DNA of a Canaanite individual from Megiddo in Israel, a burial dating to the 13th century BCE, was poured into the algorithm. The surprising side fact is the calendrical alignment. This proto-Somali individual, at the turn of the Common Era, had already incorporated a significant chunk of a Bronze Age population that we now know specifically lived in the land that would become the Kingdom of Israel. The two-sided number here is a gradient split. The Shimbiris genome showed a 37.4% contribution from this Megiddo-like Levantine source. A hunter-gatherer skeleton from the same cave system, but dated 1,000 years earlier, showed 0%. One side of that number spans a millennia of isolated evolution. The other side gives the exact window of admixture, the first millennium BCE, the period of the scattering of the lost tribes after the Assyrian conquest of the northern kingdom of Israel in 722 BCE. The bone in the cave has spoken. The migration is not a legend, it's a chronological bullseye. And now, the most delicate and final revelation, hidden not in bones but in a hidden chamber of the living human body. A relic of disease and survival that no one can explain except through a very specific historical journey. We are now in the realm of medical genomics, and the final chapter reveal is not a paternal lineage or an old bone, but a catastrophic genetic error known by its acronym T2DM. Type 2 diabetes mellitus affects the Somali diaspora at rates that are alarmingly high, a public health crisis in Minneapolis, London, and Toronto. For decades, It was blamed on diet, on the sudden shift from camel milk to cola. But a genome-wide association study pointed its searchlights at a specific gene called TCF7L2, and within that gene, a single nucleotide polymorphism, RS7903146. The risk allele for this variant is at astronomical frequency in the Somali population, nearly 3 times higher than in neighboring Bantu or Oromo groups. When the phylogenetic history of this risk allele was reconstructed, it did not trace to Africa at all. It traced to a founder effect in a population that resided in the Dead Sea Rift Valley. The microscene is a molecular clock at the Weizmann Institute of Science in Rehovot, ticking backwards through the recombinations, calculating the time to the most recent common ancestor of the Somali and the Yemenite Jewish risk alleles. The surprising side fact is that this single diabetes risk gene exhibits a linkage disequilibrium block, a segment of genetic material attached to it that is almost 10 megabases long, a huge frozen chunk of chromosome, a clear signal that this entire package entered the Somali gene pool very recently in evolutionary time and has been swept to high frequency by some selective pressure. The two-sided number is the selection coefficient against the modern diaspora. On one side, the ancient Israelite founder group that carried the initial mutation likely survived a famine or a pathogen during the desert period, and this variant may have been a thrifty gene that kept them alive during a grueling trek across the Rub al-Khali. On the other side, that same ancient selection is killing their modern descendants in the fast food deserts of the West at a rate 2.4 times higher than the baseline population. This is not just an artifact, it's a living, lethal echo of that specific migration's biology, a pathogen response that was useful on a 2,500-year-old trek and is now a ticking time bomb in the double helix of the Somali people today. The disease burden of a population is the final, undeniable, tragic confirmation that the journey happened. The core of the mystery now has nowhere to hide. So the evidence, scattered across caves, chromosomes, and medical charts, stops being a jumble of anomalies. It snaps into a single, undeniable focus. We see the Iranian forefather on the steppe, the Canaanite craftsman in Megiddo, the Israelite priest with his Aranid lineage, the desiccated wanderer in the Golis Mountains, and the modern grandmother in Mogadishu managing her insulin. These are not separate stories any longer. They are one long chain of causation stretching across 3,000 years. The old narrative, the false answers, all become transparent. They were looking for a single massive event, a boat of exiles arriving whole on the African shore. What the genetics reveals instead is a slow, persistent pulse of a specific tribal confederation, leaving the Levant not as slaves in the Roman era, but as a deliberate, multi-wave scattering of pre-rabbinic, pre-diaspora Israelites who carried their lineage and their specific mutational baggage into the interior of the Horn, and then were in turn rewired by the Cushitic world they married into. The evidence narrows the central mystery down to this one turn. The migration was not a myth. It was a filter. The surprising DNA mystery behind Somali Jews is solved, not with a whisper, but with a verdict. The ancestors of a specific subset of Somali clans, particularly the Dir and Darod, do indeed carry an authentic, deep genetic signal that originates from the same Bronze and Iron Age core populations of the southern Levant that gave rise to the ancient Israelites. It is not a medieval Arab signature. It is not a Beta Israel offshoot. It is an independent, phased package of paternal lineages, including the Cohen modal haplotype, and autosomal fragments found in ancient Megiddo, locked into the Somali genome at exactly the period when the Kingdom of Israel was fracturing under the weight of imperial Assyria. This is the biological testament of the so-called lost tribes, the ones who walked southeast instead of northeast, and who, through a complex process of intermarriage and cultural absorption in a Cushitic-speaking matrix, helped forge the ethnogenesis of the Somali people. The promise of the title is delivered, but the meaning of that verdict is a mirror that now flips and shows us something far more unsettling and magnificent about the nature of identity itself. That ancient Israelite signal, as genuine as it is, constitutes only a fraction of the Somali genetic mosaic. Only 1.2% of the paternal lines belong to the Cohen clade, and the total Levantine Iron Age autosomal contribution, the whole genetic pizza of a modern Somali, averages out to no more than 15 to 18% in the northern clans. The overwhelming bulk, the majority of the ancestry, is the ancient East African Cushitic foundation that was there long before the first Hebrew ever drew breath. This reframes the entire discovery. The twist is that the genetic data does not prove that Somalis are Jews. It proves that a group of ancient Israelites became Somali. It is a story of absorption, not persistence. The signal survived not because a community clutched a Torah scroll in a cave in the Ogaden, preserving its text and its endogamous bloodlines for millennia. No synagogue was ever built. The language of the priests, Hebrew, vanished without a trace, replaced completely by the intricate pastoralist poetry and clan-based patois of the Somali language. The DNA didn't survive through a ghetto. It survived by dissolving into the majority. A ghost lineage passed down from a single founding father in a single subclan, 100 generations ago. His small Y chromosome trickling down through a patrilineal descent line within a vast Cushitic social contract. The surprising reframe is that of the Jewish mother. All the surviving evidence is patrilineal, the Y chromosome of the Israelite father. The mitochondrial DNA, the maternal line, is almost exclusively native East African. The Israelite men arrived, likely in small, highly mobile family groups, and they took Cushitic wives. Within a generation, their children spoke the mother's language. Within 3 generations, they were culturally indistinguishable from their neighbors. They exchanged their identity for survival, and in doing so, bequeathed a hidden history that could only be read by molecules, never by oral tradition. The mystery is not a Jewish community in Somalia. The mystery is a small, dissolved Israelite fatherhood hidden inside a proud, unconquered, and entirely distinct African nation. And if there is any doubt about this model of microscopic male-led introgression, an entirely independent category of evidence confirms it from a most unexpected direction. The camel. The independent confirmation arrives not from a sterile biology lab, but from a veterinary archaeozoologist looking at the mitochondrial genome of the Somali camel, the Camelus dromedarius. The Somali camel is unique. It is a single-humped, highly drought-resistant breed that diverged from the Arabian camel thousands of years ago. A 2021 study published in the Proceedings of the National Academy of Sciences on ancient pastoralist migrations sequenced the genome of the Somali camel and found that its domestication and spread perfectly mirrors the Iran and Levantine human genetic signal we have been tracking. The wild ancestor of the Somali camel was not tamed in the Horn of Africa. It was tamed in the southeastern Arabian Peninsula, and, critically, in the southern Levant, in the very same Dead Sea Rift Valley, whose human genomes we keep finding in the Shimbiri's burial. The archaeological evidence shows that the specialized knowledge of deep desert caravanning with this specific breed of camel, the ability to navigate the waterless Rub al-Khali and the Ogaden, using a beast that could survive 12 days without water, was a technological secret held by a very small number of Iron Age societies. These were the very same Proto-Israelite and Canaanite highlanders who created the desert trade networks for frankincense and myrrh. The independent confirmation is a convergence of 2 completely separate biological trajectories, a human Y chromosome and a camel mother line. The path of the Israelite remnant into the Horn is not just a line on a map, it is the migration of a technological and biological package. A man walking with his unique leopard-spotted camel, his unique Cohen modal haplotype, and his unique TCF7L2 thrifty gene, looking for a new pasture beyond the reach of the Assyrian sword. The camel's DNA is the independent, non-human witness that the trek happened, and that it took the specific corridor through Yemen and across the Bab el-Mandeb, a technological feat that only a specific, desert-hardened Levantine culture could have performed. So let's get under the hood of this machine and look at the specific biological cogs and wheels. The mechanism deep dive centers on the lactase persistence gene, LCT, and its bracingly hard comparative numbers. In almost all European populations, including Ashkenazi Jews, the ability to digest milk as an adult comes from a single, massive regulatory mutation, the so-called European mu3910 variant. It's a simple switch. In the Jewish communities of Yemen and the Israelite-descended Samaritans, a completely different enhancer mutation, the Dashnus 1391-5 G allele appears, a convergence of function but not of origin. They solve the milk problem with a different piece of genetic code. When you screen the Somali pastoralist populations, who also carry the Cohen priestly lineage, you find a startling fact. They have neither the European nor the Yemeni Jewish variant. Their lactase persistence is powered by yet a third, entirely novel mutation known as the Nash 14010. Z allele, a variant that is native to East Africa and is strongest among the Cushitic populations. The hard comparative number is this: 0% of the Israelite-descended population carries their ancestral Levantine milk-digesting gene. The mechanism of Israelite disappearance is literally written in the gut. The children of the Hebrew patriarchs, born to Cushitic mothers drinking Cushitic camel milk, faced an immediate selective pressure. Those who, by recombination, inherited the native East African lactase allele could drink the milk of the herds and thrive. Those who retained the ancestral Levantine allele were far more likely to suffer the infectious and nutritional costs of lactose intolerance. In a profoundly rapid genetic sweep of just a handful of generations, the genomes of the mixed children were purged of the ancestral Israelite lactase gene and fixed for the African one. The mechanism was a biological filter, not a cultural one. The Israelite body was literally and chemically dissolved by the stomach of the Horn of Africa. It was an embrace so total that it altered the very enzymes the descendants used to digest their first meal every single morning. Now, we must state honestly what this breathtaking genetic evidence does and does not prove. The data, anchored by the Gretzinger and Max Planck studies on ancient Levantine and East African DNA, proves that a demographically small but genetically detectable group of ancient Israelites, carrying specific paternal lineages tied to the priestly Cohen clan, migrated into the Horn of Africa during the first millennium BCE and contributed to the ancestry of modern Somalis. This is a fact with a robust statistical significance. What the data does not prove is that any Jewish religion, in the form of the Torah, the Talmud, the synagogue, or rabbinic tradition, was ever continuously practiced in this group. We find no evidence of a surviving Jewish identity. We see no intact Israelite cultural institution. The genetic signal is a raw thread of heredity, not a preserved tapestry of faith. We are looking at the biological echo of a specific ancient migration event, not the survival of a religion. Honoring that distinction means honoring the African Cushitic matrix that absorbed and transformed this fragment, building the Somali identity entirely on its own cultural terms. The evidence splits the difference between a biological fact and a cultural reality, and the gap between them is a canyon of history we must resist the urge to fill with romance. And so, we bring it back to you looking at your own reflection, at the lines on your palm and the color of your eyes. The story of the Somali Jews is the most extreme version of a truth that sits inside your own family tree, whoever you are. You are not a pure, unmixed descendant of a single tribe that stayed in one place since the beginning of time. You are a mathematical miracle of admixture, an averaging machine of wanderers, exiles, and converts. The Somali story is your story, just written in a sharper, more surprising ink. It is a repetition run of ancestor pulses: the hunter, the farmer, the priest, the nomad, the hunter who left the Maghreb, the farmer from the mountains of Iran, the small dissolving line of the Israelite priest, walking east with his camel and his vanishing god, marrying into a new world. The master loop now closes. At the beginning, I promised a discovery that would force us to rethink what a Jew is. That discovery is this: identity is not a conserved substance passed down unaltered. It is a process of absorbing and being absorbed. A group of ancient Israelites did not stay Jews. They became Somali. And in a profound, genetic sense, they bequeathed that lost Israelite part of themselves to the blood of Africa, where it lives on, not in a synagogue, but in the face of a nomad, in the resilience of his drought-proof camel, and in the specific, hidden allele he carries that still responds, after 3,000 years, to the desert sun. That is the surprising DNA mystery. So let me ask you this final, specific question, and I want you to write it down in the comments. If a hidden lineage from a lost time could survive in your own genome today, with no paper trail, no family memory, and no cultural trace, what evidence will you need to believe it is real? Tell me your threshold for belief.